Thank you for that very kind and generous introduction. It's a great pleasure to be here. The day which I was appointed to the Scientific Advisory Board of ASF was a really proud one. It felt like I'd made it as an autism researcher, so I'm delighted to be here to talk about some of my work. I'd like to talk today about the prospects for genetic medicines. I'll start with my disclosures. I don't think any of these are relevant to what I'm going to talk with today, though. So when I started my PhD, I was presented with this simple idea. If you could understand the genetics of a disorder, it would lead you to the biology, and if you understand biology, you can develop therapy. Genetics offers an unrivaled starting point in this quest. Put simply, your DNA is set before you are born, and therefore anything which happens afterwards cannot change it. And so because it becomes before the symptoms, it makes it much, much easier to establish a causal relationship. And it was this in mind which, with the support of many in this room, embarked on this quest to try and do this in autism. This first one has turned out to be a huge success. And here, I would note largely through data provided by the Simons Foundation, we now have a list of hundreds of genes associated with autism. So this is the list of them. The size of the circle represents the number of individuals affected by each one. And this, I think, was a real surprise. You don't need to go back too far in literature to see questions about, could autism be maybe 4 or even 5 different genetic disorders? And here we are seeing that it is hundreds. All told, as we heard from John, about 15% of autistic children, if they have the right genetic testing, will end up with one of these genes being identified as a genetic diagnosis. And that group tends to be enriched for various different measures of severity. Put simply, it tends to be particularly individuals with profound autism. And so this is a group where there's ample opportunity to try and improve the lives of them and their families. So I thought I'd give us a little report card of how we're doing on this. So the good news is, on the genetics side, we're doing a good job in making solid progress. There is much, much more to do. There are many more genes to be identified, many variants to be found. Particularly on the common end of the spectrum, we've only just hit the inflection point where we start to be making good progress. But broadly, we know how to do it, the data is coming in, and the methods are working well. Things are not looking so good downstream. If we think about trying to understand the biology, despite huge amounts of work going into this, we just don't really have a clear understanding. And the fear here being, how much progress can we make without a full understanding of how a human brain works? And consequently, it's maybe no surprise without an understanding of biology that the quest towards therapies has so far been frankly disappointing all round. And so we've come to realize that there might be a way of trying to make quicker progress here for that 15% with a genetic diagnosis. Could we essentially shortcut the bit in the middle, the bit where we have to understand how a whole human brain works, and simply say, if a gene doesn't have enough of it being made or has too much being made, could you just modify that? And the logic that you've fixed the underlying problem, you might see an improvement in symptoms. Now, I think the important piece here is there'd be great benefit to the individual with that variant, but also it'd answer a fundamental question. Can you treat this? Can you make a difference? Even if you could do this just once reliably and be absolutely sure, that would give you hope that you could do it for everyone else. We start also by noticing that even in 2015, it was observed that if you started off with a single gene disorder, your chances of succeeding in making a therapy was 7 times higher. And I suspect that number's gone up a lot since 2015. We live in a time where this is starting to be possible. And this feels, to me, still a bit like science fiction, that we can actually start to interfere and change an individual's genetic factors. There are broadly 3 ways of doing this. The first one is called oligonucleotides, or antisense oligonucleotides. These act at the level of RNA, and that means that they are reversible. And I'm going to talk more about that in a second. We like the reversibility because this is a new field and we need to proceed with caution. In the middle, we have the idea that you could replace a missing gene or supplement a gene where there's not enough of it. An example here would be a therapy with SHANK3, which you can hear about at the back of the room. And then we also have the idea of actually editing the genome. This would lead to a permanent change in the DNA. And if you knew where to do that, it could be beneficial. I would note that both gene therapy and CRISPR genome editing rely on getting the product to a cell in the brain, and that remains a big challenge still. So we're going to focus on the first one of these, oligonucleotides, where we've had some more progress. When we look at the field writ large, though, we see that this can work. There are multiple disorders now where these types of therapies have had a dramatic effect on individuals treated. The problem lies that a lot of it at the moment is anecdote. So, antisense oligonucleotides. We're going to start off here by thinking about DNA. So here's DNA on the left of the screen. Now, to do something, your DNA needs to make RNA. So RNA is a single-stranded version of DNA which goes off to make proteins, and we're built of proteins. An antisense oligonucleotide is basically a small bit of RNA-like material. It is also a single strand, and that means that it can bind to one place, and ideally one place alone, in your RNA. Now, the ASO is chemically modified, and that does two things. The first thing it does is means that it really clamps onto the RNA, and that's how it has an effect. The second is that it lasts a long time. So these are often dosed every 3 to 4 months by an injection into the spine, a little bit like an epidural. So we live at a time where these have actually entered into phase 2 and beginning in phase 3 clinical trials in disorders of the brain. And I'd like to talk a bit about 2 of these, because I think they give a good idea about what might be possible. I should mention that neither of these is work done by myself, and I don't have any financial relationship with the companies which have done this. So the first one is thinking about antisense oligonucleotides in Angelman syndrome. So this is a developmental disorder. And it's caused by the absence of the protein called UBE3A. So in the middle here, we can see 2 copies of UBE3A in our genome. We have one from the mother, maternal, and one from the father. Now this is an imprinted locus, and that means that in the paternal copy, in your neurons, there's another gene called UBE3A antisense, which inhibits it. So all of the UBE3A protein comes from the maternal side. The problem in Angelman syndrome is that that mother's side one is disrupted, often a deletion. And because the paternal one is deactivated, it means that there's no UBE3A protein, and that causes the symptoms. Here, the ASO targets that antisense, and if you inhibit an inhibitor, you get activation. And so by degrading the antisense, it reactivates the paternal copy, and that leads to UBE3A protein. So here's the published, or the public results from one of these studies. There are 3 different companies working on related compounds doing this. And what we're seeing here is the table from their website showing the results of the phase 2 clinical trial. And it shows that in almost all of the things they measured, more of the patients went up than you would expect by chance. And they make a statement here that there are improvements in communication, cognition, and motor function. Now, we should take this with a slight grain of salt. This is an open label trial. That means that there was no control group. And so this is also done by report from the individuals receiving it. We should also note this has not been published or peer-reviewed. We just have to take the company's word for it. However, 3 independent companies have each moved forward with a phase 3 clinical trial in this area. Now, if we take a moment just to take this at face value, this suggests that in children, communication, cognition, and motor function can be rescued after birth. Another example is a disorder called Dravet syndrome. It causes severe seizures. Here, both copies, the one from the mother and the one from the father, lead to a functional protein called NAV1.1 because geneticists like to make things difficult. The problem here comes that one of those two copies, it can be either one, gets disrupted, and that means you have half as much protein as you need. And this issue of having half as much protein as you need is probably the most common thing we see amongst the genes associated with autism. So to fix this, we need to increase the protein, and here an ASO can interfere with the RNA, changing the splicing patterns to upregulate the amount of protein, thus improving the underlying disorder. Again, these are the results by the company on their website at the end of a phase 2 trial. And what we see here is an improvement, a fairly substantial improvement, in the number of seizures being had with the dosing of these. And they make a comment that, again, there are improvements in behavior and cognition and motor skills. All of the same caveats exist. This is not, the data are not publicly available, it's not been peer-reviewed, it was an open-label study, but still, at face value, they say that this is truly disease-modifying. If this is the case, could we make ASOs for the other hundreds of disorders there, and could we create unambiguous data that this can be done? Here I see immense potential. So this is an example of an ASO. I think this is a technology which can scale vastly. And the reason I think this is because already someone, Tim Yu at Boston, has found a way of reprogramming a therapy. So on the left here, we have an ASO for spinal muscular atrophy. This has been a huge success. It's given to thousands of people across the world and has a life-saving effect on a devastating disorder. Tim Yu met a child who had a rare genetic variant causing Batten's disease, which is a horrible neuroprogressive disorder. And 11 months after finding the variant, he was able to make an ASO, test it for safety, and get it approved by the ASO to give it to the child. And so this idea that you could do this quickly, if you know how to do it once, the next one is easier, I think is truly transformative. In fact, I think this is the first class of medicines, maybe along with mRNA vaccines and CRISPR genome editing, where this is possible, that if you can do it once, the next one is exponentially easier. And so a group of us are trying to harness this scalable potential. We see a way of taking this forward and doing it at scale. It starts off with trying to generate ASOs in vast quantities, rather than doing one at a time, trying to do it in parallel by automating the process. If we want these to scale, we need to share the data. The only way to do it is by learning by the successes, and even more importantly, the failures of other groups. This is perhaps the biggest issue. That data, which I showed for Dravet syndrome or for Angelman syndrome, it has not been shared. It probably won't be shared. We need this to happen probably in an academic setting where the data are freely available so we can actually start to learn from it. And a group of us have started working together to build a platform to do so. We need advances in analysis. A very talented student in my lab in Oxford was able to hoover up data from ASOs in patents, and from this build an AI model which reduces the number of ASOs you need to screen by a factor of 5. And we expect vast further improvements as we get more data going in. The hardest bit will, of course, be logistics. How do you get this so it's embedded into healthcare? Rather than anecdotes you read in the newspaper, how do you get it so it's your kid who has this? Now here, we see vast progress as well. The FDA have released a new program for rare therapy called the Plausible Mechanism System. And a group of us are thinking today about how would you use that system to safely regulate ASOs and bring them to clinic faster and cheaper. I've talked about ASOs and how it was possible to make one of those in 11 months, just to say that this idea of doing this at scale isn't just ASOs alone. Other genetic medicines could be done the same way, for example, genome editing. And I'd like to end just by giving this example here, and this was done at a mixture of CHOP and with colleagues at UC Berkeley, where they did the first ever base edit in vivo in a child. And the thing which was remarkable was they went from diagnosis in the first week of life to a therapy which edited this child's DNA in their liver in just 7 months. Imagine we could do that. Imagine we could use ASOs to show what was safe and what worked, and then use CRISPR and genome editing to make it permanent so you didn't need redosing. We hope that's a therapy which can be brought together in the next few years. And I will end by saying a big thank you to our MRC Centre of Research Excellence in Therapeutic Genomics, which is quite a mouthful, but they do insist that I say every single word. And thank you for listening.
Hi, great talk. My name is Crystal. I'm a PhD student working with the Seaver Center, and I had a question about like the feasibility of using ASOs for therapeutics and for all of those hundreds of genes. Is it possible that there are current and available medications that can be applied for that are used for like other diseases that are currently like already approved that can be used for some of these disorders? Or do you think the most effective and more likely to be more efficient in treating some of these genetic disorders are purely the ASOs?
Right, so with a small molecule, so it's a normal medicine, you can run vast screens of hundreds of thousands of them, but you need to measure something. And the problem we have is I don't know what to measure. Because we don't have a clear biomarker of what autism is, or we haven't identified the right subtype to link the biomarkers we have, I don't have an endpoint to run my screen. Now, I could, in theory, run the endpoint of being upregulating the gene of interest, like ADNP or SHANK3. But to do that, I would need a very, very accurate measure, and then I'd have to screen hundreds and hundreds of thousands. With an ASO, because I can predict where it goes, my chance of success is just higher. Now, in the example of spinal muscular atrophy, it wasn't just an ASO, it was also a gene therapy, and then eventually a small molecule. And so the idea that this is not, the ASO I don't think is the end of the story, it's the beginning. And once you know that something is possible, you know, if I was to go to a big pharmaceutical company and say, I unambiguously know that I can treat severe autism, profound autism symptoms in an 8-year-old, will you now do a small molecule program? I'm much more likely to succeed.
Hi, Jamal Hare from MapLight Therapeutics. I was just wondering, when you showed the slide of all the hundreds of genes, are there any that are considered monogenic for a subtype? And conversely, like, are there examples where if there is a mutation that there is no autism diagnosis?
Right, so this gets a little bit, it really fits nicely with John's lovely picture of those cups. There are some of those single gene disorders where the effect size is so vast that everything else probably doesn't matter too much. If you have the loss of that gene or a gain of function, you're going to have some kind of severe neurodevelopmental disorder. On the tail end, the sort of genes towards the end, the polygenic piece is much, much more evident. And it might increase your chance of having autism more like a 10-fold difference. What we don't have at the moment is a really clear delineation of which gene is where. The genes at the top of the list are more likely to be enough on their own. Towards the bottom of the list, they're more likely to play in the polygenic space. And autism seems to inhabit almost the perfect balance between a polygenic model and a Mendelian model. With more data, that will become clearer. Or, if we start using the therapies, if we see the therapy works, if it really was possible to make ASOs for multiple of them, actually that therapy piece would also give you an idea of the effect size. I'll always take the applause though, so thank you. I think there's another question.
Hi, thank you for doing this. I'm a parent of a child with a rare neurodevelopmental genetic condition. And as we're thinking about genetic approaches, therapeutic approaches, I guess my question would be, when there is significant heterogeneity in a disease, how do you think about scaling a specific approach? You know, ASOs are very individualized, but the DEF1 gene mutation that my daughter has over 90 variants. So how do you think about, coming up with a treatment that can essentially be scalable and effective for a broader population?
Right. So when I think about genetic medicines, I think of them as being in 3 groups. there's a group which targets the individual variant. And so that example I gave of the base edit in the liver, that was a variant-specific therapy. There's a group which targets gene-wide. So for example, SCM1A or the UBE3A, that is a gene-wide therapy, the hope being that the same therapy could be used for everyone with Angelman syndrome or everyone with Dravet syndrome. And then there's a group in the middle where you can treat a subset of the gene. And so for DEF1, my best estimate is that it's a dominant negative. I think that's still somewhat debated. But so there, an ASO type approach would try to target a common variant on the same allele as the mutation in DEF1. The hope being you could then decrease the dominant negative, allowing the wild type to act in a normal way. It would probably not be a perfect genetic rescue. In fact, it definitely wouldn't be. But it would at least show that there was rescuability in the system for you to then start thinking of things like a base edit, which might make it permanent as the technology improves. And so the hope would be to try and start at the gene and subtype level, simply because it would help the most people at any time. All right, I think we're there. Thank you.